And then next we have David Sheehan, who is the founder, president, and CEO of STOIC Bio, Inc.
Thank you. I'm Dave Sheehan. I'm the founder and CEO of STOIC Bio. I want to thank Aquilas, Mai-Fung, and Leah for putting this on. A great event. I'd love to hear all the creative ideas that are going on in terms of how to solve our environmental issues. I'm going to take a little bit of a turn. We're very life science focused, so I'm going to give you a little bit of a different product and talk about how we're changing bioproduction with Krakatoa as our product. So in the future, just happening now, disease will not be cured by taking a pill, small molecules, what that's called. It's going to be cured because we're going to be genetically modifying our cells, or we're going to be using a cell therapy that's off the shelf. And right now, that's really the future for pharma and biotech. And so we have this massive engine now, which is all of drug discovery, and it's turning towards biologics. 40% of pharma's pipeline right now is biologics. And if you look at the main areas called cell and gene therapy, there is a tremendous number of clinical trials that have kicked off. Not only that, there's already approved therapies for leukemias, lymphomas, and we're moving into solid tumor. And then there's a huge number of companies that are forming around this space. So this is really the future for our industry, which is the pharma industry. All these cells, all of these gene therapies need to be grown in liquid media. That is a requirement. The problem is we may be solving generational disease, but we're crushing the planet in the meantime. And the pharma industry is a bigger polluter than the auto industry. And that's in total greenhouse gas emissions. And the problem is these cells are grown in media that are done in large scale bioreactors. And the only way to get this very high stringency media to these bioreactors is to ship it as a liquid. That is the industry standard. Large companies, Thermo Fisher, Cytiva, Corning, they're all shipping their media around the world in liquid form. And it's really causing a tremendous problem. So the solution that we've come up with is called Krakatoa. And the name Krakatoa, if you've recognized it, it's a volcano that erupted in 1880 west of Java. It's regarded as the loudest sound heard on Earth. That's the reason why we came up with it. We think it's this disruptive of an idea for our industry. It's really transforming the biologics production market to point of use. And I want you to think about it like we're going to ship out pods in powdered form and solubilize it right in the bioproduction suite. It's like a large scale Nespresso machine for cell culture media. So the benefits of this, it's a 65% reduction in CO2 emissions or greenhouse gas emissions. The majority of it is coming from transportation, manufacturing, and end of life. Most of our production that's done for liquid media is done in single-use disposables. All of that gets incinerated. It's all sent out in single-use disposables. All that gets incinerated. What we're creating is a closed loop where we send it out in a pod in powdered form, and the customer solubilizes it right at point of use. This is important. We are the only ones right now in the industry with this solution. It is a patent pending design. We've been working on the design for several years, but it can service about 1 billion of the 1.8 billion media market. That market's going to grow to 4.2 billion. So this is just for cell and gene therapy. It doesn't really count media that's being used for vaccine development. It's not a market that we're currently going after, but it's a market that we can over time. We're very focused on the cell and gene therapy market. In terms of cost savings, to give you a perspective, we had a customer that ordered 100 liters of a specific cell culture media. They were in Cambridge, Mass. We made it here and send it to them. About $25,000. We modeled doing that in a Krakatoa, and it's less than half. Even if you count cost of the capital for that piece of equipment and amortize that over 7 years, it's still half the cost. So we're not only transforming the environmental footprint, but we're dramatically reducing the cost implications for the customer. So our go-to-market strategy and timeline here is it's really a complete solution. We have a device. It's about a little under 4 feet tall. The pods are about the size of a briefcase. You can customize it and put any type of cell culture media you can think of into that pod. We have smart software, so we create basically what's called a digital batch record, that production run that the customer can then use and put into their regulatory filings. We have a support team. Our timeline right now is Q1 2025 would be a beta launch. We have an alpha prototype already in-house. And then through the first half of 2025, work on process iterations and product iterations, and then launch in the second half of 2025. So this is something where we already have a small scale version of it, a lab bench scale, and this one is specifically for bioproduction. We have a proven and experienced leadership team. All of our team has been involved in this space, in the media manufacturing and liquid media space. We have broad experience in manufacturing GMP products. This is not an FDA approved product, but it is a GMP product because our final product, the cell culture media, is being used to manufacture a drug product. A little bit about STOIC Bio. I founded the company in 2020. It was spun off from Nucleus Biologics. If that name sounds familiar, it's because we're right next door. We manufacture liquid media right next door. Nucleus has been around since 2016. We launched a small scale version of the Krakatoa. It's called the K.5. It's a lab bench scale system. That is out there now in the market and been successful. We have 32,000 square feet between this building here and the building across the street. That's for Nucleus Biologics. STOIC is a sister company. We've got 50 employees between the two entities, and we make a variety of formulations, basically available for customization on an online platform. What's critical about it is that we're lowering cost. It's better for the environment. It's disruptive to the industry. So we're really changing the paradigm for our industry. And then in terms of financial projections, we anticipate this machine will sell for about $300,000. The pods will vary based on what formulation is in there and the cost of the components, the powder components that are inside. But this is a pretty conservative ramp. And we get to basically cash flow break even by the end of 2026. So it's a great model. It's really high margin on the pods, like 80+% margin, because we're saving so much cost in terms of the transportation. Thank you.
Thank you. And do you have a question?
This has been done for over 20 years for microbial media, right? Why is this the first time we are hearing about cell culture media? What's the secret that you have discovered?
I think the status quo is super comfortable for the companies that are in the space. And the status quo is send out liquid media. There's no reason to change that paradigm. And what we recognized as a liquid media manufacturer in the Nucleus side was we needed to change the paradigm because we needed to break this cycle that was destroying the environment. And so when we went out and looked, there were absolutely no patents that were reading on our invention when we went and originally filed for it. So I don't know why nobody ever implemented it. I think it's because they have high margin business and they like the current model and they don't care about the environment.
And have you I'm pretty sure you have tested, but do you have data benchmarking, like, gas exchange, once your product is put into an incubator alongside, other liquid media?
It's the same. We've done a lot of comparative studies. It's the same. So there's no difference. You're starting first principles with the same raw material. So it's all amino acids, proteins, everything is powdered form. It's the same whether you're making the liquid media in the customer site in a bioreactor suite, or we're making it here in San Diego.
Wonderful. Well, thank you so much,
Yeah,
STOIC
thank you.
Bio. If you want to talk to David.